My name is Jennifer Starr, and we're going to be talking about prokaryotic DNA transcription. So first, our direction of transcription for this example is going to be moving to the right, and this is because we transcribe DNA from the 5' to 3' direction, and the base pairs are read from the 3' to 5' direction. So RNA polymerase will be heading to the right during the whole process. Here's our RNA. Now this bottom strand is what we call the template strand, and this is essentially the blueprint and what will be used to read, while this top strand is the coding strand, which is complementary to the bottom template strand. So our first step in transcription is initiation. We have this promote region as well as a -35 region and a -10 box. These are basically the landing pads for RNA polymerase. And once RNA polymerase is attached, this +1 nucleotide will be the first nucleotide to be transcribed. Our next step is elongation. The RNA polymerase will unwind the DNA and create a transcription bubble, and it'll continue moving to the right. Next, we have these RNTPs, or ribonucleoside triphosphates, and these are basically the synthesizing building blocks to create the new mRNA. And RNA polymerase will add these RNTPs and create phosphodiester bonds. After about 9 to 12 base pairs are read, a sigma factor is going to dissociate from the hollow enzyme. And the hollow enzyme was attached to the consensus sequences and basically is one of the kickstarters for transcription. And our third and final step is termination. RNA polymerase is just going to reach a termination point, and it's going to have to dissociate from the template DNA. We have two different types. Our first type is row independent. So we can see a hairpin loop here. Basically, inverted repeats are transcribed, and this causes the RNA to fold in on itself, which causes a hairpin. And hairpins cause multiple adenine residues, and the adenine uracil bonds are really weak, so the RNA polymerase can't really hold on, and it falls off. Our second type is where we use Rho, and Rho binds to the RNA and moves towards the 3' end. And RNA polymerase will pause at the hairpin. And Rho has helicase characteristics, so this causes the RNA-DNA hybrid to unwind, and it all dissociates. So what would happen if we inserted more base pairs on the template DNA? So if we insert 5 base pairs in between the -35 region and -10 box, we're going to get some issues. And this is because adding those 5 base pairs will turn the -5 region into -40. And, this region is really important because this is where the RNA polymerase is going to bind to kickstart transcription. So if that is messed up, then the whole entire transcription process is messed up. And these 5 base pairs are going to code for an enzyme that produces phenylalanine. Well, phenylalanine isn't going to be produced basically because the transcription is not going to be able to be kickstarted. But in the event that an organism were to have extremely high phenylalanine amounts, especially from these 5 base pairs, this organism would experience brain damage, mental retardation, and intellectual disabilities.